Yeah, thanks very much for the kind introduction and for the invitation to come here. It's wonderful, especially since I heard we are kind of a mixed group of people, not just basic scientists, but and clinicians, but really from all walks of life, of people who might be interested in HCV. So I try to make, immunologists, as you might have seen before, tend to overload with a lot of crazy slides. I try to make it so hopefully, everyone can get something out of it. Do we have a pointer here, or most likely not? Okay, so I will try to explain where we are on each slide. So I also have to say I have to start with a little bit of a downer, I think, because if the question is T cells to the rescue, I can tell you already now, don't hold your breath. We need to come up with something better. I think the T cells might be doing it in the end, but we have to modify them at some point, and I tell you now why. So when you think about, the problem we have, it's, we all think, now since we can really cleanly take away the antigen out of the system here, which we think is really causing all the problem of the immune system that we call exhaustion. Is it just okay if we take out the virus out of the system and it will miraculously recover? Or are they really, after often, 20, 30 years of being infected, are the defects hardwired? It could be epigenetics, and we have some recent evidence for that. Are some key populations completely lost? Maybe, and so pretty much we can do whatever we want, we take it away, but there's just something missing for recovery. And there are just a few studies. That's why we'll mostly show very much data from our recent study today, because we don't know that much yet what happens on the T cell side. There are some studies from the TIMIC group in Germany, and they show that the CD8 T cells proliferate a bit better after the therapy. And there's also a certain subpopulation that expresses this, transcription factor TCF1 that are maybe kind of a source for new cells that are somewhat preserved at low level and then can give rise to new cells. But that's kind of mostly where we are. And so the big question is, after the DA treatment, if we get an improved T cell system, will that be good enough to protect us? We know that if we clear naturally HCV, there's some kind of protection. You all know it's not sterilizing or perfect, but if you clear it once, you have a pretty good likelihood to clear a second or third time. And so the question is, can we at least get to that level? And we know it's important because a lot of the patients we treat, could very easily be reinfected because they stay at risk. So, and we have seen already now that people get reinfected after the therapy. So it clearly happens, but we don't know how often are they exposed, is there some protection or not. So, we wanted to look into that and see, could there be that we have at least some partial protection that could potentially be boosted to get to a better level. So, here we are. So I introduced data today from a trial that we started a couple of years ago. And the key here is that for the first time, with this new intervention of the DA therapy, we thought it's a unique opportunity, not just for HCV, but for immunology in general, to do something new and really design a clinical trial just for the science, and not just tag on a couple of tubes to do a few studies. So the key here is, we treated overall in the end 25 patients. And from each of these patients, we got a leukapheresis product right before they got treated, and another one after treatment 12 weeks later. And we also got several passes of a liver fine needle aspirate, so we can actually also look at the site of infection. This is something really exciting to us, because we know that when you look in the blood, the immune response is not necessarily fully representative of what actually happens where the infection takes place. So now with the leukapheresis especially, and that's what the data I showed today, we have about 100 to 200 times more cells, immune cells available for our studies than we have with a regular 60 milliliter blood draw. So all of a sudden we can do a lot of things. And, this is just for the immunologists a little highlight. Usually people who have tried to analyze HCV-specific cells in chronic patients, those are the cells that are these little clouds on top. Usually you see 5, 10, 15 of these dots. And now all of a sudden, we're having so many cells and being able to enrich, we can do hundreds, thousands. We had a study published recently that did epigenetics, where we enriched from just one patient 10 and 15,000 specific cells in the chronic state, which is really unheard of. And so now, we did a whole range of studies. It's not just what my lab is doing with T cells. We look at innate immunity, epigenetics, and a lot of studies in parallel. So you will see now over the next couple of years, hopefully, a whole range of papers emerge, all from the same patients, so that we get hopefully a really complete picture of what's happening in DAA treatment. So going back to the T cells, what we really care about is mostly, what we think at least is key, is what we call T cell exhaustion. And you see here, this is just a general draft. Many people, at least in immunology, will have seen it. That if you have continuous stimulation of T cells with high antigen loads, and you have also high inflammation, and this goes on, whether it's an infection that is chronic or whether it's cancer, the T cells kind of go down a road of getting worse and worse. So they usually start expressing these what we call inhibitory T cell receptors, PD-1 and CTLA-4 being the most famous ones because they already found their way in therapy, but there are many, many others. And with that, the cells become less and less functionally and potentially even get deleted over time. And so the question is now, we take the antigen out, do they just travel back and everything is fine? When we look at this problem, the key is that, this just shows the interaction between a HCV-specific T cell and a hepatocyte is the TCR signal, we think, is what's really key here. So you need a specific signal to the cell that recognizes its antigen. And if that is prolonged, that triggers this whole thing of exhaustion. And so that's one key thing to understand, that it really mostly requires a specific simulation. It's not just a random process. It's really highly specific for whatever antigen persists. And this allows us also, in the case of HCV, a really interesting control, because as many of you know, HCV is highly variable and can escape from the T cell response. So the virus can develop mutations, and then the T cell actually doesn't see the virus anymore in the circulation. And so that means the TCR signal is gone. And so you kind of have an internal control for cells that actually are in the same infection, in the same patient, but actually do not get signal from antigen. And so this should give us, an idea of what actually happens if you do not have this continuous stimulation of the cell. So what we did is, we looked at these 25 patients. In each of the patients, we screened for T cell response. And you see, on this table, each line is a patient, and then the row through are the different hepatitis C specific T cell epitopes that we looked for. And if you see a green dot, it means that we found an epitope, and it's actually an epitope that also sees the virus. If you see a red dot, it's one that is there, but it doesn't see the circulating virus because of a mutation. So in most patients, we have a couple of these responses, and in many cases, we also have one response that does and one that doesn't see the virus. So we have really nice controls. So we then had also many other controls. You can see in these patients, we can find many other viruses. A lot of those patients have EBV, CMV, and the flu. And so in most of these, we also have other viruses, and we can so pretty much have more controls to look at what happens to responses against other viruses if we clear HCV. Is it a specific effect, or do we see also something global here? So we then, because we had so many cells, we just made a couple of really big panels, and this is like 37 different markers we looked at. You don't need to know exactly each one what they are, but the keys, we looked for things that show whether a cell is activated, whether a cell has these inhibitory receptors or other inhibitory effectors, some markers that show you where the cells are going, kind of that's the homing markers, are they going to the liver or lymph nodes, etc., cell death, and we define what kind of memory subtype they are. So you get a pretty broad picture of how those cells look and how they change over time. And this is the kind of graph we will now see in the core of the talk quite a few times. So I explained a little bit. I did it on a Mac, didn't come out fully well. I hope you can still see it. So it's called a spider graph. So you have in a circle all these 37 markers, and it's like a fingerprint almost of each cell, what they express and what they not express. It's just from 0 to 100% expression. So if it's really far out, you have 100% expression. If it's nothing, you have 0. And so you see right away that several of these markers are are highly expressed, especially at like 1 to 2 o'clock. You see several of the inhibitory receptors are really high, and we kind of knew that to be the case. You see a lot of activation that's like at 5 o'clock. There are markers of activation, and there are a couple of others that are really high. And then this is before we started treatment. And so now we look at what's happening after treatment. That's the blue line. And you do see there are quite some changes, especially at the 5 o'clock range. The activation is gone, so it makes a lot of sense because the cells don't see the antigen anymore. It has been removed. And then we do see some memory phenotype markers come up. That's kind of was reassuring or made us hopeful, because it seemed to indicate that from being classical exhausted T cells, we see a bit more memory T cells, which is what we would see in a recovered infection. And then you see some change, but not a lot, it seems, on these inhibitory receptors, like at 1 to 2 o'clock. But actually there was more going on, it seems, because this is just showing that the cells still mostly express these markers. But if you look relatively how much do they express, then you see on the right now that, for PD-1 and CD95, for example, that while they still mostly express the marker, the expression levels go actually down by quite a lot. We're talking about almost a log range, and that makes definitely a difference. So there's certainly on this level some improvement. The next question then was obviously what happens to the mutated epitopes, and you see here pre and post together, and you just see that these cells don't change at all. So it kind of confirms that it's really a highly specific process that we have here, that only the cells that got the specific TCR signal are changing over time, whereas those with the mutated epitopes do not show any change. And same with the other viruses, including if you take the whole CD8 T cell population, they just look before and after exactly like exactly the same. So it's really only limited this change to the HCV-specific T cells that do see antigen. So then the next question was obviously, is this as good as if you naturally clear? And so we took a group of patients and we tried to be as similar as possible, same genotype, no co-infection, and we also studied them after acute infection about the same time frame, the same number of weeks, 20 to 24 weeks, and the other patients had last time seen HCV because it was then cleared. And so we looked into those patients, and now you see, in green, the resolvers, and in blue, the post-treatment chronic patients. And you see already here that they're quite distinct in many ways, especially, on the top right, these inhibitory receptors are already, when you go by a percentage, not the same, and the memory phenotype is not the same. And when you look individually more carefully, you see actually quite significant differences still, for like PD-1, the most important marker maybe, and some of the transcription factors that are really relevant here. So the answer here already is they don't go all the way, clearly. So we then used all the markers, and you can this is called a Tisney plot, and it kind of projects all these 37 markers in a two-dimensional space. And so if a cell is in the same spot than another cell, it means the whole pattern looks kind of very similar, and if they sit somewhere else and far away, they're very different. And if you just look at the top 4 plots, you just see first the exhausted T cells, like, the preserved ones in the chronic patients before we treat them. And they sit all in this upper left corner, and they're very red because they express really high levels of PD-1. And then when we treat them, they move down kind of in the middle, and you see lower expression levels of PD-1. And when you compare that to their kind of, the comparator of the cells that did not see the antigen even before they were treated, they still are a bit further down and have a bit less PD-1. So they don't even reach fully kind of the cells that had this early escape and kind of, lost within a few weeks of being infected, lost their antigen. And this indicates to us, and there's other data we have now, that actually the time of exposure to antigen makes really a difference. If you have it, within a year or so, treat or the antigen just disappears by the mutation, the cells might be more likely to recover than after 10, 20, or 30 years of chronic infection. So that's obviously gives us a bit of headache because a lot of the patients, have it for quite some time. And then you see obviously on the right, the resolvers are on a different planet kind of altogether. So there's really still some way to go all for these cells where they should go. So with that, I conclude at this point that after cure, we do see phenotypic changes that indicate really there's some kind of recovery and some kind of reversal of the exhaustion. But it's not, it's definitely not complete. In the blood, we don't see changes in these control epitopes, whether it's the mutated epitopes from HCV or any other cells. So it's really, really specific for HCV and really requires, this TCR stimulation. And then, as I said before, we don't get full recovery. And so now obviously we need to go further from this, and learn more details. And just going to show or tell you a couple of things we're doing. So the first one is obviously, phenotyping is one thing. What's the function? And one thing that is underway right now is with Paul Glenneman in Oxford. He actually vaccinates some of these patients, not from the same cohort, but after similar treatment, vaccinates with a T-cell vaccine they developed. So we're going to see at least in vivo in the patient, if you give them a challenge with HCV antigens, are those cells better able now to respond? Because I did the same trial in chronic patients a couple of years ago. And in chronic patients, it was they were dead in the water, and you could vaccinate as much as you wanted to, and nothing happened. So the question is now, we vaccinate, will it be better? And obviously, we are doing functional assays. We luckily have these huge numbers of cells, and I show you this almost done, but I just show you an example here to give you an idea. It's pretty representative. This is kind of an interesting patient we saw several years ago during the acute phase of infection, followed for a couple of years, didn't want to be treated, and then when this trial came up, the patient said, "Yeah, now it's time," and I get treated. So you see on the left, these are functional assays. It's always in this each square you see 4 different plots. The left 2 are without stimulation of antigen, and the right ones are stimulated. In the top one you see TNF alpha and interferon gamma, and the lower one you see CD107A, that's a marker for cytotoxicity, and again interferon gamma. And so you see in the acute phase, you see, on the upper left, you see some function of all these cytokines is there. And we know it's not very good in acute, generally in HCV, and especially not patients who become chronic, but there's some of the function there. And then you see below, an epitope that mutated early, has definitely more function already. So again, indicating that when antigen is lost early, the cells remain more functional. But then when we look in the chronic phase, that's the middle panel on top, there's still actually CD107A, that's the most preserved one, but the cytokines are actually almost absent. And if you look onto the cured cells, there's a little bit of recovery of TNF alpha. And we have seen now the cohort is almost complete. So we do see actually, in contrast to what was published by others before, but we have these large cell numbers, we do see some modest recovery. But I can already tell you, we have been doing now the acutes as well. Who have recovered. That there's a massive difference in the functionality of these cells. So they're really not there yet. So then obviously that brings us to the next question. We have done really complex microarray, actually RNA-seq experiments on these cells, both from the liver and from the blood, whether we can identify molecular targets that we could potentially modulate to push these cells to the next level, and we hopefully find something, because it will definitely be necessary to either vaccinate these patients or find some other modulation of these T cells to make them fully functional. We are working on these liver samples. We have some early data showing that it's a little bit different in the liver. It most likely has to do with the fact that, in the liver we see most likely constant little changes in the viral sequence in the chronic phase, but that's nothing that's really relevant for this. And we see a bit of a different phenotype that will be interesting to follow up. And then the big question, HCV, and I wanted to show it today, but we still need to do the final experiments. What about the T cells, the CD4 T cells? Because we all know those are kind of the real key. Without those, there's no control of HCV. And we did this in the cohort I just described to you. I didn't show the data because there's actually not much to show. We enriched from 200 million cells, and we usually find no or a couple of dots of CD4 T cells before treatment, and it doesn't really change after. When it does change is when we treated a cohort of co-infected people, in the acute phase. And in those, you can actually salvage the CD4 response. It actually comes even back a little bit on treatment. And so it seems that, again, and Heiner had this shown many years ago that, in the old interferon era, that there might still be potentially some kind of benefit of treating early, but we'll see. I'm still not sure that those cells will do it, but I can at least say we can save them when we treat them early to a certain degree. So with that, I just want to acknowledge my lab. Actually, this Pierre Tonnerre, did I not even write him on? He's actually the one who did. Yeah, most likely I stole his slide, and so he didn't put himself on. So it's Pierre Tonnerre in my lab who pretty much did all of this work and did a really great job on this, and all the collaborators from the consortium. With that, if there are any questions, I think we have maybe a minute or so. Oh, discussion later? Oh.